today I'm going to talk about intimate interactions that occur between male and female gametes that happen post-mating but pre-zygotically. And we're going to focus today on those.
Behind the mic.
What?
Behind the mic.
Behind the mic, okay. And we're going to focus today, better? Okay. We're going to focus today on plants. So here we have a nice cross-section of a flower where we can see the male and female components right next to each other. So when we're talking about male and female interactions in plants, we're talking about the instance where male gametes or pollen lands on top of the stigmatic surface and must traverse all the way down through the style to the base where the ovary is located and the female gametes or ovules are. And just like in plants, we can invoke female choice via active pollen rejection in the style, so this component here, the female reproductive tract. And it's this choice via genetic and biochemical control mechanisms that regulate fertilization against the potential for heterospecific pollen, and that can lead to a rejection or a non-rejection phenotype. So ultimately leading to fertilization. So essentially we're talking about reproductive isolation via pollen rejection in the female reproductive tract or the plant style. But we're going to ask the question, what is the genetic basis of interspecific pollen rejection, but from the female perspective? So what are the factors that are contributing to a response for pollen tube rejection in the female reproductive tract? And in order to do that, we're going to use the species in the clay that contains domesticated tomato, so Solanum lycopersicum, and one of its wild relatives, Solanum pinelii. In addition to being able to generate interspecific crosses between species in this clay, we also have a number of well-characterized pre- and postzygotic barriers. In addition, we have this really interesting phenotype called unilateral incompatibility. So unilateral incompatibility, or UI, is essentially a post-mating pre-zygotic reproductive barrier where pollen rejection occurs in this style in one direction of a cross, but it's successful in the opposite direction. So in this instance, pollen from species A is placed onto the stigma of species B, and we generate a rejection phenotype, or we consider this to be an incompatibility phenotype. But in the alternative cross, when pollen is placed on species B, we do not see a rejection of pollen in the style of the alternate species, so we consider this to be a compatible cross. And in some work done over 20 years ago, 3 QTL were mapped to chromosome 1, 3, and 12 that contributed to the expression of unilateral incompatibility. However, this mapping population is genetically complex in order to address the genetic basis of interspecific pollen rejection from the female perspective. So we're going to address a question about the contribution of loci towards interspecific pollen rejection, but without the complexity of this F2 mapping population. So in order to ask this question, do we see evidence for epistasis for stylar factors? We're going to do two things. We're going to isolate the individual effects of these candidate loci on the expression of this pollen rejection phenotype, and we're also going to examine the effects of 2 candidate loci on the expression of UI. Okay, so we're going to use this powerful genetic tool that's been developed in Solanum, called integration lines. Essentially, we just have a genetically defined homozygous region that's been donated from Solanum pinelii into the background of Solanum lycopersicum. And this is found across all 12 chromosomes in Solanum lycopersicum. And just as a visual representation, the UIQTL that were identified about 20 years ago on chromosome 1, 3, and 12 do overlap our introgression lines. However, to note, the UIQTL for chromosome 3 actually overlaps 2 different introgression lines, which we're just going to call introgression line 3.3 and 3.4. So in order to address both the individual and the combined effects of these loci on this expression of a pollen rejection phenotype, we're going to generate double introgression lines. And so by doing that, we're just taking parental single locus introgression lines generated in an F1 individual, and then screening genetically for individuals which contain both introgressions on 2 different chromosomes from the donor parent, in this instance, Solanum pinelii. And we genotype these individuals with multiple species-specific markers. And so the data that I'm going to show you today is only with 3 crosses. They all include chromosome 12, and then it's with either chromosome 1 or the 2 different introgression lines on chromosome 3. And then in order to actually quantify the heterospecific rejection of pollen, we're going to take Solanum lycopersicum tester pollen and place it onto our nil or DIL genotype, so our single or double introgression lines, and quantify or measure the pollen rejection phenotype. And so this is just a little bit of eye candy of what that actually looks like. The top here, the stigma, we've fluorescently stained this, and these nice bright blue lines are pollen tubes, where we get this really nice rejection response about 3/4 of the way down the style. Okay, so just to orient you to how I'm going to display the data, we can think of the top of the graph here as being the top of the stigma, where pollen is placed at this position. It must grow through the style in order to reach the ovary located at the base, or the female gametes. So if we see a rejection phenotype, we would see pollen growing possibly to this region, maybe 3/4 of the way, and we would infer some sort of rejection occurring around here. And then alternatively, we would have no rejection, so we'd see pollen tubes reaching the base of the style and growing into the ovary or the ovules, so essentially allowing fertilization to occur. Okay, so if we want to look at the individual effects, we're just going to essentially screen pollen rejection in our single parental introgression lines. And when we do that, we do not see the rejection of heterospecific pollen, so there's no unilateral incompatibility. And I'm just plotting here each of the style genotypes on the X, and then these are just confidence intervals. So we see no significant difference between the parental lines on the expression of this phenotype. However, when we do one pairwise cross between chromosome 12 and 1, we do not see a significant difference between the double introgression line and our parentals. So essentially, there's no difference in terms of pollen tube growth or pollen rejection in this cross. However, when we look at the combination of chromosome 3 and 12, we do see a response. We do see pollen tube rejection, and so essentially we can infer that this phenotype is actually happening, or we do see the expression of UI. And that these double introgression lines with chromosome 3 are significantly different than any of the parental lines. And then if we think about the data a little bit more, we see variation for chromosome 3 on the expression of UI. So here is just a representative image for each of these crosses in terms of their pollen rejection phenotype. And just in case you forgot, we have 2 introgression lines which have a partially overlapping region. So that's what we're calling 3-4 introgression line and 3-3. So we're basically inferring that there's at least one genetic factor on chromosome 3 found within the overlapping region, because we do have some sort of response in terms of pollen rejection, but that response is even stronger in the other double interaction line. So we basically see a weak response or an early acting response and a late acting response and a much earlier acting response in terms of the rejection of pollen tube growth happening about halfway down the style. That's a pretty active response in terms of generating this, in terms of stopping pollen tube growth into the ovules or the female gametes. So essentially we're saying there's two factors on chromosome 3 which contribute to the expression of unilateral incompatibility, or the active heterospecific pollen rejection. So I hope that I've shown you that we do actually see evidence of epistasis for these styler factors. First, we showed that there are no individual effects of these loci on the expression of UI, and that when we combine chromosome 3 and chromosome 12, we do actually generate this unilateral incompatibility response. So essentially we can infer that there's epistatic interactions happening. And then furthermore, we were able to dissect the original UI-QTL into two genetic factors because of the partially non-overlapping regions where we saw an early and late acting response. So, in summary, I think we can talk about female choice from a plant perspective against heterospecific pollen, and that we have epistatic interactions among greater than one locus required for this response. So the natural next question is, do we see even more complex or three-way epistasis, epistatic interactions between these loci for chromosome 1, 3, and 12? And we are actually in the process of generating these triple integration lines and to genotype and to phenotype for this response. And with that, I'll thank all the Moyle Lab members and many undergraduates that have helped with this project, as well as some funding, and I'll take some questions.